Welcome to the Cannabis Plant, a Clinical Perspective Cannabis Morphology Learning section. Cannabis is a dioecious annual plant assigned to the Cannabaceae botanical family. Its pharmacopoeial name is Cannabis inflorescentia, which refers to the dried inflorescences which contain the active pharmaceutical components of cannabis. Some common colloquial names for cannabis include marijuana, hemp, weed, ganja, and sensimilla, which is a slang term for cannabis grown without seeds. The Cannabaceae family not only contains cannabis, but also humulus, which contains a popular plant used in most beer brewing known as hops. There has been a long disagreement between whether cannabis is a monotypic or polytypic species. The monotypic argument was that the cannabis gene pool was heavily influenced by human agriculture, which then split the cannabis species into sativa and indica. In 2005, new genetic evidence indicated that the split between indica and sativa actually predated human intervention, and most botanists now agree that there are two main species of cannabis plants. Indica plants are thought to be native to Asia and tend to grow short with thick stems and broad, deep green leaves. They have short flowering cycles and grow sufficiently in cold, short-seasoned climates. Sativa plants, thought to be native to Europe, have longer flowering cycles, fare better in warm climates with long seasons, and usually grow taller with light green, narrow leaves. In 1930, a third subspecies of cannabis was identified by Russian botanist Dmitry Januszewski as Cannabis ruderalis. This time, it was not a result of unique physical characteristics, but rather unique traits in the plant's flowering cycle that identified it as a possible subspecies. Januszewski noticed that while most cannabis plants begin to flower as a result of the changing available sunlight, ruderalis plants automatically flowered between 20 to 40 days after sprouting. He noted that ruderalis is most likely representative of a wild-type hybrid blend of the various cannabis gene pools that survived as a feral escape, rather than as a direct descendant of the now long-extinct ancestral cannabis population. The argument for or against the existence of Cannabis ruderalis as a third species of cannabis has little to no effect on the clinical indications of cannabis, as this subtype is rarely used or cultivated for medical purposes. It is also important to note that hemp is not a distinct cannabis species. Hemp is Cannabis sativa, which contains less than 0.3% THC. Hemp represents just one of the many varieties of Cannabis sativa and Cannabis indica containing various percentages of active cannabinoids. Cannabis is a herbaceous annual plant that is primarily dioecious, which means that male and female flowers occur on separate plants. Because the cannabinoids are primarily found in female flowers, when cultivating cannabis for medical purposes, all female plants are used, and males are typically not allowed in or near the grow area to avoid accidental fertilization. However, as often happens, nature finds a way. Female cannabis plants can become monoecious and grow male and female flowers on the same plant in different locations. Very rarely, cannabis plants can also become hermaphroditic and produce bisexual flowers. Typically, this occurs in response to changes in the photoperiod during flowering, poor environmental conditions, such as temperatures that are too high, physical stress on the plant, such as pruning during the flowering stage, infestation of mites and insects, or incorrect fertilization processes. The female plants tend to grow shorter and more robust than their staminate counterparts, but the terminal height and degree of branching of the plant varies widely based on genetic and environmental factors. Cannabis plants are typically fertilized through wind-borne pollination, and they crossbreed indiscriminately across genotypes, which makes the separation of specific cultivars of cannabis very important in order to maintain the medicinal composition of cannabinoids within the plant. In its original form, cannabis has probable origins in Central Asia, the northwestern Himalayas, and China. Native growth occurs in open habitats along riverbanks, bottomlands, and hillsides. Humans have dispersed the plant worldwide over the past 10,000 years through migration, trade, and modern cultivation. Cannabis is not indigenous to North America. It was brought here through trade and cultivation and is now a naturalized plant in more than 30 states at altitudes from 0 to 2,000 meters. It grows primarily in fertile, moist farmlands, waste areas, fallow fields, and open woodlands. Cannabis is cultivated both in and outdoors for recreational and medical purposes. Indoor cultivation is by far the most common growing method in the United States, as it allows for better control of crop outcomes. The cannabis plant has many non-recreational uses that have largely been forgotten due to the focus of cannabis as an illicit drug. Hemp seeds are high in protein and essential fatty acids. Hemp stems provide one of the strongest natural fibers known and have been used historically to make sailcloth and rope. Hemp seed oil can be used not only as a nutritional supplement, but can also be used to formulate biofuel. And of course, the unfertilized female flowers can be used to create pharmaceutical compounds. The different uses of cannabis are representative of the wide variety of plant types that grow within the species. Taller, more fibrous plants are typically used for seeds and stems, whereas the bushier, flowering plants are used for pharmaceuticals. Additional information on these topics can be found in our Growth, Manufacturing, and Testing Learning module. The pharmaceutical compounds found in cannabis, known as cannabinoids, are found in the upper leaves, unfertilized flower heads, and flower bracts of the unfertilized female plants. There are no cannabinoids present in the roots, seeds, or stems of the cannabis plant. Cannabinoids are encased in tiny glandular trichomes located on the leaves of the plant. Some trichomes are large enough to be seen by the naked eye, but many are microscopic and give the plant a frosted, dew-like appearance. These trichomes manufacture and contain the active cannabinoids made by the plant. Some are bulbous, stalked, and glandular, while others have calcium carbonate crystals at the base and form a hooked, rigid appearance on the surface of the leaf. The sticky, cannabinoid-rich substance produced by the trichomes is commonly referred to as cannabis resin. It is important to understand that within the cannabis species, there are many different chemovars which are grown and cultivated for their different cannabinoid composition. Certain plants are very high in THC, some are high in CBD, and still others have a more equal ratio of both THC and CBD. Rampant crossbreeding has largely negated a species-specific composition, so each chemovar must be individually evaluated for its unique cannabinoid profile when being grown for medicinal use. Changes in environment, time, and stress on the plant can also change the cannabinoid profile, making it even more challenging to produce consistent plant-based medications for patients.